Hello all, my name is Pradeep Kiran Jangampalli Adi. I am working as a research assistant professor at Texas Tech Health Science Center, Lubbock. Today I would like to present a brief topic on small molecule drug discovery and possible therapeutic development in Alzheimer's disease and Tauopathies. Introduction of Alzheimer's disease. It's a progressive fetal neurodegenerative disorder characterized by memory loss followed by a general loss of cognitive and other brain functions. The cognitive changes with AD tend to follow a characteristic pattern, beginning with the memory impairment and spreading usage and visuospatial defects. In the elderly, nearly 50 to 60% of the cases of dementia are due to Alzheimer's disease. Incidence is 70% in 65 to 69 years old and 40% in 95 years and above. Most cases are sporadic, some are familial. We know Alzheimer's disease is a progressive neurodegenerative disorder characterized by memory loss followed by a general loss of cognitive and other brain function. Changes first begins in the enteral cortex and hippocampal. As the disease progresses, damage spreads to the areas of cerebral cortex that controls the language, reasoning, sensory processing, and conscious thought. Stages of AD classified into preclinical, MCI, mild to moderate, and severe AD. Possible sequences of AD events, which includes the formation of amyloid plaques, formation of neurofibrillary tangles, inflammatory reactions, oxidative damage, loss of synapse, and neurons, in particularly cholinergic neurons, and dementia and progressive neuronal loss and cerebral atrophy. Tick diagram represents amyloid hypothesis, amyloid plaques formation. APP, amyloid precursor protein, was cleaved by the beta secretase, alpha secretase, and gamma secretases. These secretases cleaved at different regions of APP and leads to the insoluble and soluble fragments of A beta. These amyloid fibrils deposit make A beta aggregates, then leads to the damage the neurons. Amyloid plaques, A beta 1 to 40, more neurotoxic, predominant in parenchymal deposits. A beta 1 to 40, less neurotoxic, predominant in vascular deposits. Exit in vivo at concentration ratio of 1 is to 10. Increase in the ratio leads to the early onset familial AD. No, neurofibrillary tangles are the intracellular collection of twisted protein threads. Tau containing phosphate molecules binds to the microtubules and appears to stabilize them. Neurofibrillary tangles NFTs are the intracellular aggregates of hyperphosphorylated tau protein that are most commonly known as the primary biomarkers of Alzheimer's disease. Their presence is almost found in numerous and other diseases known as tauopathies. Formation of neurofibrillary tangles or hyperphosphorylation of microtubule associated protein tau, causing it to aggregate and group into insoluble form. This hyperphosphorylated tau protein, also referred as a PHF. The presence of the mechanism of tangle formation is not completely understood, though it is typically recognized as a tangles are primary cause factor of neurodegenerative diseases including AD. Genetic consideration, APP gene, chromosomal 21, early onset familial AD, Presilin1, PS1, PS2. PS1 gene is located at chromosomal 14 encoded with S182. PS2 gene is located at chromosome 1 encodes with STM2. ApoE gene located at chromosome 19 late onset familial AD has a 3 alleles ApoE4 as a strong association with AD. Status of Alzheimer's disease is characterized by cognitive assessment, clinical dementia rating CDR, Minimental Statement Examination MMSC, neuroimaging MRI, SPECT. Approved therapies for Alzheimer's disease, cholinesterase inhibitors, and NMDA receptor modulation. Cholinesterase inhibitors includes like dinopizil, gelatamine, rivastigmine, and tarcan. These drugs provide some side effects. And NMDA receptor modulation like memantine, non-competitive NMDA receptor antagonists. These drugs also provide some side effects when treating the patients. Most of the drugs was discontinued in clinical trials. Experimental therapy for Alzheimer's disease mainly focus on current drug discovery and developmental program leading approaches which includes the inhibition of A beta production, enhance the A beta clearance, enhance the A beta degradation, and finally inhibition of A beta assembly. Are some alternative disease modification strategies which includes the RNA interference. It is a process of mRNA degradation induces by hybridization with a short sequence of complementary RNA, there are thereby forming a dsRNA. It is useful in early onset FAD caused by mutations in the APP gene and preselin1. Currently, there is no disease modifying therapies approved for AD. A number of promising targets and therapeutic strategies for the treatment of Alzheimer's disease are under actively participating in drug discovery programs. My research focus on finding the novel molecules ligands to treat Alzheimer's disease through computational based screening. Next, treating and testing small molecules in neuronal cell cultures and AD mouse models. I designed and tested small molecules for amyloid beta 4042 and phosphorylation of tau in Alzheimer's disease. As you know, the accumulation of amyloid precursor protein fragment from base amyloid beta 42 leads to the formation of plaque that disturb the synaptic communications in neurons. Abnormal phosphorylation of tau protein leads to the breakup of microtubules and generate neurofibrillary tangles in AD. These resulting in the loss of cargo axonal transportation cause the neuronal cell death and neurofibrillary tangles in AD neurons. In my first year postdoctoral research focused on screening the small molecules to target base inhibitor ligands. In the study, I constructed a pharmacophore base with pepstatin ligand based screening through computational drug discovery and molecular modeling. We found three potential ligands in our molecular docking analysis of base inhibitor ligands. We tested three base inhibitor ligands for APP processing and A beta inhibition in AD neurons. We used biochemical, molecular, and transmission electron microscopy and immunoblotting analysis. We studied ligand 1 protective effects against A beta induced synaptic mitochondrial toxicity in mouse neuroblastoma cells that express mutant APP. We found physical interaction between the ligand 1 and base 1, and this interaction decreased the abnormal APP processing and reduced the base activity and A beta 40 and 42 levels. We found increased mitochondrial biogenesis, mitochondrial fission, synaptic activity, and reduced the mitochondrial fission. Based on these results, we found that ligand 1 reduced the A beta induced mitochondrial synaptic toxicity in AD neurons. Mechanism of selecting small molecule for base inhibitor ligand, I selected aspartate protease inhibitor as a reference ligand which specifically binds to the aspartic acid. In the complete base domain, there is a specific aspartic protease inhibitor which specifically binds to the aspartic acid in the base. So I screened the complete zinc database to find a significant molecule which specifically binds to the aspartic acid. So based upon the molecular docking studies, I selected the top binding ligands to the base inhibitory. The base protein has a multiple domains which includes a signal peptide, procatalytic, transmembrane, and cytoplasmic domain. Catalytic domain contains two aspartic protease active sites of the motif, which is responsible for the catalytic cleavage of A beta 40 and 42. Based on the concept, we first did the molecular docking studies with pepstatin as a reference ligand and prepared a grid pharmacophore. This pharmacophore recognized as a pepstatin related ligands. While training the ligand molecules, we did with the we trained the zinc database to find the pepstatin related ligands. These ligands further trained the molecular docking studies to find a potential ligands. Based on the molecular docking studies, we selected top 3 molecules. Further studies based upon the molecular docking scores and tested in cell culture model. We tested ligand 1 in 4 N2A cell cultures. After treating ligand 1, we collected a protein lysate to run the Western blots. As we see, the clear full-length APP C99 and C83 significantly decreased in ligand-treated mutant N2A cells. We also verified other biomarkers, mitochondrial fission and fusion synaptic markers were significantly improved in mutant APP ligand-treated N2A cells. The complete results was published in Human Molecular Genetics 2000 and the patent issued with the ligand 1 molecules in 2024. My second study focused on screening the small molecules to target hyperphosphorylated tau. Tau is a phosphorylated protein contain nearly 80 to 85 serine, threonine and tyrosine phosphorylation sites. Hyperphosphorylation leads to the imbalance in tau kinases and phosphatase activity. Hyperphosphorylated tau is reported to be an important contributor to the PHF formation and neurofibrillary tangle formation in AD. As we see isoforms of tau, some conserved regions located at 275 to 280 and 306 to 311. Many reports studied in these conserved regions unreported. But my focus, there are some other phosphorylation sites may be participated in Alzheimer's disease. After I searched for ligand small molecule which specifically binds to the serine and threonine based binding ligand as a reference molecule through database and selected a small molecule which specifically target the serine threonine phosphatases. Later, I cross-verified these phosphorylation sites are involving in kinase activity through NetFast tool. They are significantly involving in kinase activity in AD. Later, I constructed a pharmacophore grid with reference ligand which specifically binds with serine and threonine and make a model to pharmacophore. After pharmacophore screen, with the zinc database, we found some potential ligand which specifically binds with serine and threonine. After molecular docking studies, we selected ligand 1 for further studies. These all results were published in Cells MDPI. In vivo validation of small molecule p-tau inhibitor. In this present study, we rationally selected and validated a small molecule for p-tau that binds to the phosphorylated tau at serine 285, which is responsible for GSK3 beta and CDK5. Tyrosine 44, which is responsible for DURK1, which caused the hyperphosphorylated kinase upregulation in AD. We found the interaction between the ligand 1 and key phosphorylated sites and decrease the pH of tau, DURK1 and GSK3 beta levels. We also found the increased mitochondrial biogenesis, mitochondrial fusion and synaptic activity and reduce the mitochondrial fission in ligand 1 treated mutant HT22 cells. Based on these results, we cautiously conclude that tau ligand reduces the hyperphosphorylation in AD, helps to maintain the neural structure and function with a therapeutic function in AD. This slide shows the MD simulations of 3 best ligands with tau molecule. Based on the MD simulations, we found ligand 1 shows the best molecular simulation to test further. In this slide, table 1 shows the molecular docking studies of the 3 best ligands with the tau molecule and the ligand 1 shows the best docking score. We validated ligand 1 in cell survival and cell viability tests. The result shows that ligand 1 treated HT22 cells showed the protective nature in presence of mutant tau H222 cells. These are the immunoblotting studies of p-tau in H222 cells. We validated the p-tau inhibition in phosphorylated proteins, mitochondrial dynamics, mitochondrial biogenesis, and synaptic proteins. All the results shows that ligand 1 has some protective effects on p-tau inhibition in presence of mutant tau. We also validated the p-tau ligand with immunofluorescence studies. We validated the p-tau and DRP with immunofluorescence analysis. The result shows that p-tau and DRP decrease the immunofluorescence in presence of mutant tau H222 cells. My third study focused on anti-aging and p-tau inhibition properties of DDQ in mutant tau expressed H222 cells. Our lab developed a small molecule called DDQ has shown the CYP3 upregulation and p-tau inhibition in AD mice. To confirm that p-tau interaction of DDQ and CYP3 upregulation with creep-mediated mitophagy actions in AD neurons, we conducted immunoblotting and immunofluorescence analysis were conducted and validated. Result shows that p-tau and total tau proteins are significantly reduced in DDQ-treated mutant tau H222 cells and increased MAP2 levels were observed. Anti-aging proteins like CYP3 and CREP3 levels were increased in DDQ-treated H222 cells. Mitophagic proteins were decreased in mutant tau H222 cells and increased in DDQ-treated mutant tau H222 cells. These observations strongly suggest that DDQ has a anti-p-tau and anti-aging properties via CYP3 overexpression and increased mitophagy proteins. The Western blot result shows that p-tau and total levels were significantly increased in mutant tau H222 cells and decreased in DDQ-treated H222 cells. MAP2 levels were decreased in mutant tau condition and significantly increased in DDQ-treated mutant tau H222 cells. And ApoE levels were increased in mutant tau condition and decreased in DDQ-treated mutant tau H222 cells. And the CYP3 and CREP protein levels were significantly decreased in mutant tau condition and increased in DDQ-treated mutant tau H222 cells. We also verified DDQ p-tau inhibition and aging properties by conducting the immunofluorescence studies. The result shows that DDQ decreased the p-tau immunofluorescence intensity in mutant tau H222 cells treated with H222 cells when compared with the mutant tau H222 cells. CYP3 levels were increased in DDQ-treated H222 cells when compared with the mutant tau H222 cells. The mitophagy markers PINK1 levels were restored in DDQ-treated H222 cells when compared with the mutant tau H222 cells. The results was published in Mitochondrion in 2020-24. My another study focused on testing the DDQ-treated hippocampal neuronal cells shows the improved neuritic outgrowth and synaptic branching. In this work, we examined the protective effects of DDQ on neuronal process and synaptic outgrowth in differentiated H222 cells. To investigate the DDQ characteristics, we used the biochemical, molecular, and Western blotting and immunohistochemistry were used. Neuritic outgrowth is evaluated through the segmentation and measurement of neuronal process. Our Western blot studies was confirmed that DDQ shows the p-tau and total tau were significantly reduced and increased MAP2 tubulin and synaptophysin and vascular acetylcholine transporter VACHT. These findings conclude that DDQ may have some potential neuronal developmental characteristics. This Western blot results clear that DDQ shows the significant p-tau inhibition and VACHT protective upregulation in presence of mutant tau condition. Conclude that DDQ may have some neuritic protective characteristics. In this slide, we can clearly observe the H222 cells neuritic outgrowth in the presence of DDQ. We validated a MAP2 tubulin and VACHT and other biomarkers in the recently published paper Pradeep Kiran et al. 2025. These slides represent the mutant tau H222 cells and DDQ treatments of mutant tau H222 cells. Neuritic outgrowth was significantly improved and differentiated in presence of DDQ was observed. The complete results was published in Neuronal Regeneration in 2025. To the best of my knowledge and current understanding of AD pathologies involves the various hypotheses such as cholinergic, amyloid, p-tau, inflammatory, oxidative stress leads to the abnormal mitophagy in AD. My research discoveries of selective inhibitors, dual targeted inhibitors, modulators, covalent inhibitors of p-tau and A-beta may provide some meaningful therapeutic awareness for AD. These small molecules also helps to decrease the oxidative stress, mitochondrial protection and decrease the aging properties with CYP3 upregulation and ApoE downregulation. My research goal is to provide some insightful and prospective drug development with the clinical applications of novel AD drugs. Thank you.